Okay, can somebody please confirm you're hearing me?
We can hear you.
Great, thank you. Why isn't this working? Oh, there it goes. Sorry, I'm trying to call up our schedule here and my computer is a little slow for some reason. There we go. Just share the screen here. All right, you should be seeing my screen now. Today is July 21st, and we should be finishing chapter 3 and beginning chapter 4, prokaryotic and eukaryotic cells. I think we started chapter 4 last time. There will be a lab today at the normal time for the labs. Let's go to chapter 4. Can you guys hear that music in the background? If it doesn't stop, I'll shut my window.
I don't hear any music.
All right, we had talked about the different structures you can find in the prokaryotic cell. Any questions about any of that? If not, let's start talking about the different structures of prokaryotes. Some prokaryotes have a glycocalyx, which is secreted from the cell. The glycocalyx is outside the cell and is usually sticky outside the cell wall, and it can be relatively thin, like in this picture here, where the glycocalyx is the pinkmost layer, or reddish pink layer, I guess you'd call it. Or it can be very thick. If you remember, I think it was last time when I was showing you the capsules, it can be as big as the cell, sometimes even bigger than the cell. Can you guys hear that music?
I don't hear anything.
Okay, good. Those aren't real big, but there, these are bigger. So in this one right here, the capsule is much bigger, which is a glycocalyx, is much bigger than the cell. And sometimes they can be very large. There are 3 types of glycocalyxes. There's a capsule, which is neatly organized, and it has more things in it than just water. If you can see the glycocalyx in the light microscope, it's a capsule. Any questions about any of that? So there are glycocalyxes that you cannot see in the light microscope, and the main molecule of these glycocalyxes are just water. We call that a slime layer. It's an unorganized layer. It's loose, and it's mostly water, which is why it doesn't show up with the light microscope. You can see the slime layer if you use an electron microscope, but you cannot see it in the light microscope. I wonder why you can't see that with the stains in light microscope. It might be because it's mostly water and it's easily destroyed. And then the third type of a glycocalyx is an extracellular polysaccharide, which you only find in biofilms. We'll talk a little bit about biofilms later in the term. That's when the bacteria, when it's growing, puts out a glycocalyx, which then connects with all other bacteria, which adds to the glycocalyx. And then you have these cells in a mass of a biofilm that we call a biofilm that has cells in it, and each of them makes, or many of them make the glycocalyx, and it makes a structure around all the cells. And it's very difficult to kill cells in a biofilm, and that's why we'll talk about it. Any question about any of that? All right. So there are three types of glycocalyxes. If you can see it in the light microscope, it's a capsule. The next structure we're going to talk about is the flagella. Flagella is plural. A singular one would be flagellum. They're a whip-like structure, allowing the cell to move. So if the cell can swim in a liquid media or a aqueous solid, then it has a flagella. The flagella is mostly outside the cell wall, but it does have a portion that's inside the cell wall, and it does have a portion that's actually inside the cell membrane. Flagella is made up of protein, and it has a name, flagellin, and the different chains of this protein, flagellin, make the flagella. Any question about any of that? You don't need to know the different types of bacteria, flagella. They have different arrangements. You only needed to know it for answering the lab question. So I won't ever test you on, is this a peritrochus or a monotrochus bacteria? So don't memorize, I'm just showing it to you. Okay, if a cell is motile, then it has flagella. When cells are motile, they can swim, and people call it running or tumbling. Running when the cell is moving in the same direction. Tumbling is kind of, well, like a ball tumbling. The movement of cells we call taxis. Taxis is the movement toward or away from a stimuli. So I guess that's not the movement of cells, it's the movement of cells either toward or away from a stimuli. There's two types of taxis. Chemotaxis, where cells are stimulated by a chemical, and they can move either towards that chemical or away from it. And then phototaxis, shown here, where in this case the cells are moving away from the light. So they're moving this way. I can tell because the flagella are that way. Usually, photosynthetic cells move towards the light. So this must be a non-photosynthetic cell. Any question about any of that? Taxis is extremely important for bacteria because if the bacteria has taxis, they can move to a more favorable environment, and they're more likely to survive in a more favorable environment. Now there's another type of movement caused by axial filaments or a flagella, but it's a special flagella, and we call it an endoflagella or an axial filament. It's only found in spirochetes or, spirochetes. The flagella is anchored at one end of the cell inside the cell wall, and the flagella is inside the cell wall entirely. So here, I don't know if you can see it attached to the cell here, and it wraps around the cell. And this is an artist's rendition where the flagella rotates around the cell. And when we look at this, let me blow this up a little bit. When we look at this, you can see the light pink being the cell wall, and the cell wall surrounds the axial filaments or the endoflagella, which is inside the cell wall. Any question about any of that? And the artist is trying to show you that the light pink is the cell wall, the endoflagella is inside the cell wall. The endoflagella cause the cells to have a characteristic corkscrew-like movement, which I'll try and show you. Which only the spirochetes move this way. It allows them to swim. Looks like we're going to get a commercial to begin with.
Google Fi, a phone plan by Google. Save big on the Moto G family. Shop the latest phone.
Where the cell, let me blow that up, can change its direction. And not only did it change its direction, meaning it's rotating the flagella and going that way, rotating the other way and moving that way, but it actually changed its direction by, what is it, 90 degrees? Nope, I'm not that far enough. Go back further. Right there. And that tells you how limber the spirochetes are. They're not a rigid cell wall. There's one that's moving this way. Anyways, any questions about the endoflagella? The movement like this allows the endoflagella to actually burrow through a mucous membrane and maybe through a human tissue as well, as long as the tissue isn't strong, like it can't burrow through the skin, but it can burrow through the mucous membrane and through, what would it be, soft tissue, maybe inside your muscle. The next structure I want to talk about is fimbriae. It's the hair-like or yellow structures on this cell. They extend from the cell wall. They're shorter and straighter than flagella, and they're not used for movement. They are used for attaching to surfaces, so not for swimming. And some pathogenic bacteria have fimbriae, which then use the fimbriae to attach to our cells. Right, any questions about fimbriae? The pili, also called the sex pili, is a little longer than the fimbriae. In fact, we can see, if I can blow this up, let's see if I can do it from here. Still not showing it well. There, now we can see it. This cell does have fimbriae on it. It's these hair-like structures here. which are light blue. This cell does not have fimbriae, and that is the sex pili or pili, and it's much longer than the fimbriae. It's also hollow. The sex pili is a structure that actually allows this cell to skewer another cell like this cell here, and then the sex pili acts as a winch and then can bring this cell closer together, meaning it brings the two cells closer together. They don't have to be really, really close for the sex pili to act, because the sex pilus is singular, is pilus, pili is plural. It's hollow, and then it can allow a plasma to leave this cell and then enter that cell. So the sex pili is used to transfer DNA from one cell to another. Any questions about any of that? We'll talk a little bit about the sex pilus later in the term when we're talking about bacteria genomics. All right, moving on to the prokaryotic cell wall. This is a specific structure found in most prokaryotes. It's complex, semi-rigid structure, and it's responsible for giving the cell its shape. So if you have a cocci, the reason why the bacteria have the round shape is because the cell wall is round. And then if you have a rod, the reason why it's a rod is because the cell wall is a rod. All right. Now let me state that the spirochetes, shown here, they do have a cell wall, but it's not really rigid, at least in the spirochetes. In the spirals, it is rigid, but in the spirochetes, it's not rigid. But in most of the bacteria, it is rigid, semi-rigid. The cell wall protects the cell membrane and can prevent osmotic lysis. So lysis occurs when water moves into a cell, like when you put red blood cell in a bucket of water, water will move into the red blood cell, expanding the red blood cell, expanding this, sorry, I'm drawing a blank here, not the cytoplasm, the plasma membrane. Okay, until the plasma membrane bursts, and then that'll kill the cell. Well, that would happen in bacteria too. And when water moves into the cell, the plasma membrane will expand up into the cell wall. And then the plasma membrane cannot expand any further. That prevents water from moving into the cell. And that keeps the cell from expanding and then bursting the plasma membrane, killing the cell. So the cell wall can prevent osmotic lysis. Note that the cell wall is located outside of the cell membrane, and it is considered a part of the cell, even though it is outside the cell membrane. And the cell membrane, you might recall, is the portion of the cell that determines what gets into the cell and what moves out of the cell, meaning it's the one that's the gateway determining what moves into the cell and what moves out of the cell. But the cell wall is external to the cell membrane, but it is considered a part of the cell. The glycocalyx, which is outside the cell wall, is considered external to the cell. The cell wall is mostly made up of peptidoglycan, at least in bacteria. In Archaea bacteria, the cell wall is not made up of peptidoglycan. And then in eukaryotes, which have a cell wall, which we're not talking about, the cell wall is not made up of peptidoglycan. So peptidoglycan is only found in bacteria. And that's a little bit oversimplified, but we're going to go with it for this class. Let's talk a little bit more about peptidoglycan. It's a polymer of a repeating disaccharide. So disaccharide is made up of two monosaccharides. The two monosaccharides are N-acetylglucosamine, or NAG, and N-acetylmuramic acid, or NAM. And you can use either name to call it. What's making up peptidoglycan, NAG or NAM, which probably mean as much to you as N-acetylglucosamine. But here we can sort of see, it's better to look over here maybe, that peptidoglycan is repeating units of two monosaccharides, NAM, NAG, NAM, NAG, NAM, NAG, meaning they just repeat one after the other. And it's one row upon row in a plane, many different rows of this disaccharide. And then underneath this row, we have another layer of NAG and NAM peptidoglycan. And that's what I meant by layers. And then another layer of peptidoglycan under that. And in the cell wall, we have many layers of peptidoglycan. Even when you're talking about gram-negative cells, which have less peptidoglycan than a gram-positive, you still have many layers of peptidoglycan. Okay, I don't know how many layers in gram-negative cells, but it's more than just a few. Something like 20 to 100 in gram-negative, and over 100 in gram-positive. Maybe over 1,000 in gram-positive. Peptidoglycan, besides having the NAG and NAM repeating disaccharide, has short peptide links. Let me shrink that a little. Connecting the chains of NAG and NAM. And here you can see a short peptide, and that's linking this NAG and NAM chain with that chain, although not directly. What that peptide is doing is it's binding the peptide running horizontally from NAG and NAM here, and NAG and NAM here. And then this one is running across. So it's connecting the NAG and NAM chains together. And these short peptides is what gives peptidoglycan its name. Peptidoglycan means short chains of peptides, peptido, and then glycan means sugar, and NAG and NAM are sugars. So peptidoglycan, that's its name. Any question about that? The peptides holding the NAG and NAM chains together are extremely important. And in fact, that's what penicillin inhibits. When a cell wall is putting down its peptidoglycan, the penicillin gets in between the NAG and the NAM chains and then prevents a peptide from linking the NAG and NAM chains together. And so you only get one penicillin molecule here. This chain won't be held together, and that'll make a gap in the cell wall right here where the NAG and the NAM aren't held together. And having a gap there, the plasma membrane can protrude out of this gap. And then as more water comes into the cell, this plasma membrane can protrude out of the gap even further and further, sort of like a balloon being blown in a box with a hole in it, the balloon will start protruding out of the hole in the box. And eventually enough water will move into the cell that the cell membrane will burst. And that's actually how penicillin kills prokaryotic cells, at least gram-positive cells. Any question about any of that? All right, as we stated, when we're looking at bacteria cells, we have two types, gram-positive shown here, which have a very thick peptidoglycan layer, the purple, and then underneath that's a thin cell membrane layer, which is actually a bilayer of phospholipids. And then many layers, there's only 3 shown here, but it's actually many layers of peptidoglycan, consisting of chains of NAG and NAM held together, and then there's many of them going that way, and then many of them going this way. There are some other molecules in the gram-positive bacterial cell, mainly teichoic acid, running this way, which kind of just helps to hold the peptidoglycan together. And then there's lipoteichoic acid, which is teichoic acid with a lipid on it. Blow that up. And so this portion would be the teichoic acid portion, and it just kind of holds the peptidoglycan together. The lipoteichoic acid anchors into the cell membrane, and the lipid portion of the lipoteichoic acid is inside the cell membrane. Any question about the gram-positive cells? I should mention that teichoic acid can help ions move either through the cell wall or, well, into the cell wall or out of the cell wall. I guess that's what I'm trying to say. So teichoic acid, the ions can use the teichoic acid to move down or up, whichever way they're moving in the cell wall. However, ions do not need teichoic acid to move in the cell wall because ions are soluble in water, and water can move right through the cell wall. Any question about any of that? There can be polysaccharides in the gram-positive cell which provide antigenic variation. So one strain of a bacteria can have a polysaccharide on it, which then can be recognized by the immune system, and that's what we mean by antigenic variation. Whereas another strain either has a different polysaccharide or no polysaccharide, and then the immune system can't use that to mount an immune response to. So an antigen is just something that our immune system recognizes as foreign and then begins an immune attack against it. And because the molecules of the cell wall are the outermost molecules of the bacteria, these molecules are used by the host immune system to mount an immune response against. And any molecule that the immune system recognizes as foreign, we call an antigen. Any question about any of that? Here we're seeing the gram-negative bacterial cell wall. It does have a peptidoglycan layer, but the peptidoglycan layer is much thinner than in the gram-positive. Excuse me.
Bless you.
Thank you. Hopefully you didn't get too much of that sneeze. The gram negative peptidoglycan layer is much thinner than the gram positive peptidoglycan layer. There's also another portion of the cell wall of the gram negative cells, which is actually lipid or mostly lipid, and it's on the outside of the cell wall, and that we call the outer membrane of the cell wall. If you look at it, the outer membrane of the cell wall looks very similar to the cell membrane. And in fact, this bilayer is composed of the same molecules as the bilayer, this portion of the bilayer, sorry, the innermost portion of the bilayer of the outer cell wall is composed of the same molecules as the cell membrane, meaning mainly phospholipids shown here and here. And then the outer layer of the cell wall does have phospholipids in it. And that's probably the main molecule, but it has another lipid that we call lipid A. And lipid A has an O polysaccharide attached to it. So the round structure, the blue round structure is the lipid A. The O polysaccharide makes the hair-like structure. And together, when we put these together, we call it a lipopolysaccharide. The important part of this is lipid A. And why it's important is lipid A is found in gram-negative cells that have a cell wall. And lipid A is an endotoxin. Toxins are something that are always bad. They're toxic to us, why we call them a toxin. And whenever a patient has an infection with a gram-negative cell, they will be exposed to an endotoxin. Any questions about any of that? This will be the first time we're talking about toxins in this class. We will be talking about toxins 2 other times. And so they're an important topic that we'll come to time and time again. So be expecting quiz questions about toxins. For now, we're just going to say toxins are something that are bad and they're toxic substances to us. Let's see. There's a few other molecules in the cell wall of the gram-negative cell. The main one I want to talk about is a porin protein. The porin protein provides a channel in the outer membrane of the cell wall, and this channel allows molecules to go through the outer membrane, either to enter or to leave, and that allows molecules to enter the cell that the cell wants, as well as it allows molecules to leave the cell that the cell does not want. For example, glucose and all amino acids cannot get through the outer membrane of the cell wall. And it's because this bilayer prevents glucose and amino acids and many other molecules from moving through the outer membrane. However, the cell does want glucose and amino acids, and they can move through the porin protein and then get through the outer membrane of the cell wall. Now, glucose and amino acids cannot move through the cell membrane, but the cell makes a transport protein to move those molecules through the cell membrane. The difference between a transport protein and the porin protein is that a transport protein essentially only allows one molecule to move across the cell membrane through the transport protein. And if it's moving into the cell, it moves into the cell, but there can be other proteins the cell doesn't want, and it'll move them out of the cell with the transport protein. So the transport proteins are very specific. Now, if you know anything about medicine, we do have drugs which can mimic the substrate that's being moved by the transport protein. So the transport protein is not 100% specific, but it's really high, like 99% specific for that one molecule. And that's all it moves. The boron protein moves a whole slew of different molecules through it, like glucose and fructose, and all the 20 different amino acids can move through the boron protein. It's just a channel protein that allows many different things to move through the outer membrane of the cell wall. Any question about any of that? The boron protein is specific though. It doesn't allow all molecules to move through it. And the only one we're going to talk about that the boron protein doesn't allow to move through is penicillin G. Penicillin G cannot get through the outer membrane of the cell wall, and it cannot move through the boron protein. So penicillin G cannot get in the peptidoglycan of the cell wall of the gram-negative cell and then damage it. And that's why gram-negative cells are not susceptible to penicillin G. Any question about any of that? Let's talk about a few other molecules you can find in the gram-negative cell wall. You can find a lipoprotein. And this lipoprotein will have a portion inside the cell wall, and then a portion either in the outer membrane of the cell wall or in the cell membrane. And in this case, it looks like this one is both in the cell membrane, spanning the cell wall and in the outer membrane. Any question about any of that? So you'll note we do have a few extra molecules in the gram-negative cell wall than we have in the gram-positive cell wall. Let's see if I've talked about everything here. In the gram-negative cell wall, you can have lipoproteins, lipopolysaccharide, which the one we're talking about is the lipid A, and the O polysaccharide being the lipopolysaccharide. And then the phospholipids, which is the main molecule of the outer membrane. And in the lower bilayer, it's almost all of the molecules of the outer membrane. Obviously, the boron protein goes through the lower portion of the outer wall, outer membrane of the cell wall. You probably don't need to know about the periplasm, but that's the name we call for where the peptidoglycan is in the cell wall. The outer membrane of the cell wall can protect the cell, the gram-negative cell, from more than just penicillin G. I didn't mention that one. It can also protect the cell from complement proteins and antibiotics. Well, that would be penicillin G. Complement protein binds to the cell that the body recognizes as foreign, and then the complement protein can help defend the body against the cell. And if you have a gram-negative cell with this outer membrane, the complement protein cannot bind to it. The outer membrane does make it more difficult for phagocytes to phagocytize the bacteria. So it makes it more difficult for phagocytes to mount an immune response against the bacteria. And then the proteins in the gram-negative cell can give some antigens, such as when we talk about E. coli 0157 and then H7, the 0157, where do I have that? It's from this O polysaccharide on the lipopolysaccharide, which is attached to the lipid A. Any question about any of that? So 0157, H7 are two antigens on this E. coli. And if you don't know, this E. coli is a human pathogen. You don't want it in your gut because it's different from normal E. coli, which is found in your gut. And I've already talked about lipid A and porins. Any question about the gram-negative cell wall? If there's no questions, let's talk a little bit about the difference between the gram-positive cell wall and the gram-negative cell wall. The gram-positive cell walls have a thick peptidoglycan layer, giving the cell a rigid structure. The gram-negative cell walls have a thinner peptidoglycan layer, and they're less rigid. I wouldn't say they're terribly pliable like a spirochete, but they're less rigid. The gram-positive cell walls have teichoic acid in them. There's no teichoic acid in the gram-negative cell walls. The gram-positive cells do not have an outer membrane in their cell wall. The gram-negative cell walls always have an outer membrane with lipid A in it if the cell has a cell wall. Any questions about any of that? All right, you don't need to know this, but you might have noticed the way I worded that. If the cell is gram-negative and has a cell wall, then it has an outer membrane. However, there is one family of bacteria which does not have a cell wall, and it stains gram-negative in the gram stain. Okay, and obviously that doesn't have a cell wall, doesn't have an outer membrane. You don't need to know any of that, but I just was pointing out that my wording was in gram-negative cells that have a cell wall, then they have an outer membrane. Any question about gram-positive and gram-negative cells? All right, as we've discussed, the cell wall determines the gram stain. The crystal violet iodine crystals form in the cell and the cell wall. And in the gram-positive cells, the peptidoglycan is so thick that the crystal violet iodine crystals do not leave the thick peptidoglycan layer, at least if the gram stain is done correctly. In the gram-negative cells, the crystal violet iodine complex washes out of the thinner peptidoglycan layer. And then the alcohol in the decolorizing steps destroys the outer membrane. Let's go back here. The outer membrane is mainly lipids, and so the outer membrane will be destroyed by alcohol, which will also destroy the cell membrane. Where was I? Oh, and then the gram-negative cells, instead of being clear in the gram stain, they take up the safranin and then appear pink. Okay? Whereas the gram-positive cells retain the crystal violet iodine, and so they retain the purple color. And then this slide is just showing you a comparison of gram-positive and gram-negative cells. You only need to know the top portion, thick peptidoglycan in the gram-positive, thin in the gram-negative, teichoic acid present in gram-positive, absent in gram-negative, the outer membrane absent in gram-positive, present in gram-negative, lipopolysaccharide, virtually none, we're just going to say none in the gram-positive, present in the gram-negative. Any question about any of that? All right, if there's no questions, let me mention that there are some cells which have atypical cell walls, meaning cell walls which are not like I just showed you. Or a gram-negative like this, or a gram-positive, where do I have that? Gram-positive like that. There are some cells which are atypical. The first I want to mention, which I think I've already mentioned, is the archaea. Everything that we've talked about with the cell walls applies only to bacteria. The archaea generally have cell walls, but they are not made of peptidoglycan. They are made of another disaccharide. You don't need to know the name, pseudomurine. And the archaea do stain as gram-positive and as gram-negative. And once again, the gram-positive archaea have a thick cell wall, whereas the gram-negative archaea have a thin cell wall. And because it's thick and thin, determines whether crystal violet iodine complex can wash out of the cell wall. Any questions about the archaea? Generally speaking, in this class, we won't be talking about the archaea, also called the archaebacteria, because none of them are human pathogens. And the focus of this class is on human pathogens. So the archaea are important for microbiologists, but for you guys, they're just not as important as the bacteria. Another atypical cell wall of bacteria is the family of mycoplasmas. This family of bacteria is atypical because the cells lack a cell wall. Curiously, the mycoplasmas have sterols in their plasma membrane. They don't have cholesterol, for example, but the sterol is very similar to cholesterol. And as you remember, humans have cholesterol in their plasma membrane, and the cholesterol gives the plasma membrane a greater strength. And the sterols in the mycoplasma also give the plasma membrane greater strength. Any question about any of that? So the mycoplasmas do not have a cell wall. When we use the gram stain on them, will they stain gram positively or gram negatively? Anyone going to take a guess? You got a 50-50 chance here. You should be able to figure it out too. Nobody's going to take a guess. All right. The mycoplasma stain gram-negative because they don't have a cell wall. So the crystal violet iodine complex washes out of them. And then they're stained by the safranin. So they're gram-negative. The other atypical cell walls are the mycobacterium, and they're atypical because they have mycolic acid in their cell wall. Mycolic acid is a lipid, and the mycobacterium have a thick layer of mycolic acid near the outer portion of the cell wall. The mycobacterium are gram-positive, and they are stained gram-positive, but they're an atypical gram-positive cell. I don't have a picture. Maybe I can go online. I don't have one in this lesson. Someplace I do have a picture of the someplace I do have a picture of the mycobacterium cell wall, but it's not in this lesson, so I don't know where it is. I'm just going to Google to get there. There's a good one. You can't see this too well. Maybe I can get it better this way. Let's just go back here. We'll live with this. Maybe I can blow it up. Not really. I'm not seeing it there. Let's go here. Anyways, as you can see in the cell wall, it has another membrane. That's not a gram mycobacterium. I'm not finding my picture. Let's go back. Go back again. Here we go. So number 2 is the mycolic acid. And as you can see, this is the cell wall, 4 being the peptidoglycan, and it's a fairly thick layer of peptidoglycan. And then outside of the peptidoglycan, there's a few other molecules, 3 which you don't need to know about, polysaccharide. And then 2 is the mycolic acid, a very thick layer of the cell wall. And this is lipid, and that prevents water-soluble molecules to either enter the cell or to leave the cell. And that allows the cell to retain its water because water will not usually leave the mycolic acid. Anyways, the point is the mycolic acid makes an additional layer of the cell wall, which makes the bacteria have different properties from other gram-positive cells, giving them atypical gram-positive cells. The mycobacterium stain with the acid-fast stain. The acid-fast stain can get through the mycolic acid, and then they retain that dye in the mycolic acid. And so the cells will stain positively for acid-fast. And then other cells that don't have the macolic acid will stain negative for the acid-fast stain. And we talked a little bit more about that in the lab, and I think we'll talk about it a little more in a future lab. Any questions about the atypical cell walls? All right, let's move on. You don't need to know the different terms, but you should know that you can damage the cell wall of the prokaryotic cell. And I've actually already told you one way. With penicillin, this antibiotic inhibits the peptide bridges connecting the different NAG and NAM strains, NAG and NAM strings together in the peptidoglycan, and that puts a hole, even if very small, in the peptidoglycan, which allows the cell membrane to protrude out of that hole. And you can use penicillin to damage the cells. You can also use the enzyme lysozyme, which digests the peptidoglycan. A lysozyme is an enzyme that you can find on your skin or in the tears of your eye. And our body produces it to actually help prevent bacteria from getting established in us and then causing an infection in us. So this is something, an enzyme our body makes to help defend us against bacteria. Anyways, you can use the lysozyme and penicillin to damage the cells and then get a wall-less prokaryotic cell. And you don't need to know the different terms. So you saw that the gram-negative bacteria are not as susceptible to gram-positives to penicillin G due to the presence of the outer membrane. That doesn't mean that the gram-negative cells are not susceptible to other antibiotics. As long as you have an antibiotic that can penetrate the outer membrane, that antibiotic will work on gram-negative cells. Streptomycin is an antibiotic that can get right through the lipid bilayer of the outer membrane of the cell wall. So streptomycin works on gram-negative cells. And then I've also mentioned that ampicillin, a very early member of the penicillin family, I think it was the third or the fourth member of the family to be discovered, can damage gram-negative bacteria. It's only penicillin G and penicillin V which cannot damage, well, there's other members too, but these are the two that cannot damage gram-negative cells, mainly penicillin G, the first antibiotic discovery. All right, any questions about the cell wall? No questions? If there's no questions about the cell wall, let me turn to the cell membrane. The cell membrane, as we've discussed, is a bilayer, and it's mainly made of the phospholipid molecule, where the fatty acid portion comes inside, and then the phosphate group faces outside and inside the cell, making a lipid bilayer. And it's this lipid bilayer that determines what can either enter the cell or leave the cell. There are other molecules in the plasma membrane, mostly proteins. That's probably the only molecule we're going to talk about. And there are three types of proteins. Peripheral proteins, which are on one side of the membrane or the other, what's called peripheral. It's on one side of the membrane or the other. Any questions about any of that? There are also integral proteins and transmembrane proteins. Both of these proteins span the entire membrane. The only protein we're going to talk about, which I believe is an integral protein, is the transport proteins. And these proteins specifically move one molecule either into the cell or one molecule out of the cell. And we have a different transport protein to move different molecules. Like we have a transport protein to move fructose, and we have a transport protein to move glucose. The same transport protein cannot move both of those molecules. Any question about any of that? All right. When we're talking about the plasma membrane, it's important to realize that it is a fluid. And it is about the same fluidness or the same consistency as olive oil. And if you know, you put olive oil in the refrigerator, it becomes a solid for anyone who's ever done it. But at room temperature, olive oil is a liquid, but it is a thick liquid. And it's hard to imagine that my hand here, which you're looking at, is held together by cells, which are held together by the cell membrane. And the only thing holding my hand together is the cell membrane, because I'm an animal. And that cell membrane is just a fluid. That always amazes me, that a fluid is holding my hand together. Okay. Now, the fluid mosaic model states that in this fluid, the molecules can move around. The proteins can move around. They may not be fixed in place. They may be, we'll talk a little bit about that. But some of them can move. And in A and P, if you've taken that class, some of you know that when receptor proteins bind, they will move together before they're brought into the cell. So when the receptor protein binds to its ligand, the receptor proteins will move together and then be brought into the cell, and then the message will be brought to the cell. The point is that those proteins have to move in order to function. Well, we have proteins in the cell membrane which also move about. The only proteins that are fixed in place are the proteins which are either bound to the intracellular matrix, in which case, let's say this was bound to the intracellular matrix. If it's bound to the intracellular matrix, then this protein is fixed in place and it'll stay there. Any question about any of that? Oh, I guess I forgot to mention, not good. It's either bound to the intracellular matrix or to the extracellular matrix, like proteins in the bone. So we have a cell inside a bone. Well, this protein can be connected to the extracellular matrix of the bone, and then that protein will not move about in the membrane, but it's fixed in place. And it is important to realize that the phospholipids are not non-motile. The phospholipids can obviously rotate around And then they can also move laterally. I think I mentioned that that's how water can sneak across the cell membrane. And that is, initially water will be up here, but it can't move through the lipid bilayer. And what it does is the water just waits for a phospholipid to move out of the way. And then the water molecule moves into the bilayer, the upper portion of the bilayer, but it can't move through the next layer of phospholipids. And so the water just stays there inside the plasma membrane. And what happens is the water just waits for this molecule to move out of the way, and then the water molecule can move into the cell. Okay, and that's how water can move through the cell membrane when it is not using a transport protein. Now, obviously, cells that need water to move through it, like the roots of a plant cell, they will make proteins and put it in the cell membrane. And then that protein will move the water from outside the cell to inside the cell, assuming there's more water outside the cell. Okay. All right, any question about any of that? The plasma membrane shows selective permeability. What that means is that some molecules can move through the plasma membrane, but others cannot. Large molecules cannot move across the plasma membrane without the assistance of a transport protein. So that would be something like a large carbohydrate or a large protein. Can't move through the cell membrane without the help of a transport protein. Ions do not move through the lipid bilayer, or if they move very slowly. The reason is an ion is a charged molecule, and the charge does not want to be in this lipid portion of the bilayer. It'll be repelled from this region. Lipid molecules, on the other hand, are soluble in the bilayer. So lipid molecules can move right into the cell or right out of the cell. They can move into the cell membrane because they're soluble in it, and they can move inside the cell. But if you think about it, most cells, like a blood cell, is in the blood, which is mostly water. You're not going to have a whole lot of lipids outside the cell. But if you do, like you eat a cholesterol-rich diet, for example, you'll get cholesterol in your blood, and then that cholesterol will come and move right through the lipid bilayer of your cell. So lipid molecules can move through the plasma membrane. Small molecules can also move through the plasma membrane. The two most important that we're going to talk about are oxygen and carbon dioxide. Oxygen, by diffusion, can move right through the cell membrane. And why? Because oxygen is small and it's a polar, non-polar molecule. Is that right? Yeah, oxygen's being attracted to oxygen. So that'd be a non-polar molecule. And anyways, oxygen can move right through the lipid bilayer. Carbon dioxide, the same thing, can move outside the cell. Carbon dioxide is made inside the cell, so it'll have higher carbon dioxide inside the cell. And by diffusion, carbon dioxide will then move right through the cell membrane and then out into the cell or out of the cell. All right, any question about any of that? All right, the plasma membrane also has specific enzymes or proteins in it that you may not find elsewhere in the cell. For example, in bacteria, the plasma membrane has the enzymes for the cell to make ATP in aerobic respiration. I think we talked about that. Or are we going to talk about it? I don't remember. In another lesson, if we haven't talked about it, in a future lesson, we will talk about it. I think it is a future lesson for you guys. That in bacteria, as well as archaebacteria, the plasma membrane has the enzymes for making ATP in aerobic respiration. For photosynthetic bacteria, the pigments of the bacteria, pigments are the light-trapping molecules. They are found in the plasma membrane, and they're actually found in the folds of the plasma membrane. So right here, the plasma membrane is here, but it's not a straight line. The plasma membrane, actually, let me see if I can blow this up. Actually comes in here, and then it folds in here. Coming out here, folds in here. Comes in here, folds in there. Comes in here, folds in here. So it's both on the outside, and then there's an inner fold, which the inner fold is connected. Let's see if I can draw that. Hopefully I have this. So it's not just like this, plasma membrane of these cells. It's more like this. There's a blow up, and then we have a folding of the plasma membrane. Not doing so well on this one. All right, you got that? Which we're trying to show here. And these have the molecules in these folds for trapping the pigment. And these we call chromatophores or thylakoids, similar to what you find in a eukaryotic cell, which you find what is that called? Thyloids? Whatever. In the chloroplast. But you don't have chloroplast in a bacteria, but we do have the folding of the cell membrane. And it's in the cell membrane, in the folds, that we have the photosynthetic pigments. The plasma membrane can be damaged by different molecules, such as molecules of alcohol, quaternary ammonium compound, which I think that's one of the detergents that they now use in the hospitals, and then polymyxin antibiotic. Polymyxin B is an antibiotic which can put holes in the plasma membrane of the cell, or holes in the gram-negative outer membrane, or both. And that's how you can damage the plasma membrane. So the movement of materials across the plasma membrane is critical to cells. There are two processes that molecules can use to move across the cell membrane. The first is a passive process where molecules move down a concentration gradient. For example, there's more oxygen outside the cell than inside the cell, and oxygen can move from outside the cell to inside the cell, where the oxygen is moving down its concentration gradient. Any questions about any of that? It's a passive process. It's passive because the cell doesn't use any energy to move the molecule across the cell membrane. There are also active processes where a molecule can be moved into the cell against its concentration gradient. And how the cell does that, it will use a transport protein to move a molecule from outside the cell to inside the cell. And if it's an active movement, that means it's moving against its concentration gradient. So if you were to eat a high rich glucose diet, and then you were to digest almost all of the glucose, most of the glucose would be in your cells. So the glucose concentration inside your cells would be higher than outside your cells. Let's say the lumen of your intestine, because most of the glucose has been digested. However, glucose is a molecule that the cell is going to want, and so it has a transport protein to move down to the last glucose molecule outside the cell. Will move it inside the cell. And that movement is an active process against its concentration gradient. It costs the cell 1 ATP molecule to move one molecule across the cell membrane against its concentration gradient. And the cell uses the energy from ATP, meaning ATP will be broken into ADP, and inorganic phosphate, releasing energy. That energy will then be used to move that molecule, glucose in this case, into the cell against its concentration gradient. Any questions about any of that? All right. The passive movement down a concentration gradient can happen in 3 different ways. The passive movement can be facilitated diffusion, where a solute moves from one side of the membrane to the next down its concentration gradient. So oxygen moves from areas of high concentration to areas of low concentration down its concentration gradient. And oxygen is moving by simple diffusion. Oxygen is a solute being dissolved in the water of the blood, for example, and in the water of the cytoplasm. Okay. The second way that a molecule can move by a passive process is by facilitated diffusion. Once again, here it's a solute that's moving down its concentration gradient. For example, if you were to eat a high fructose diet by eating a lot of fruit, you'll have a lot of fructose in your gut. And how you digest that fructose is you make a protein, and it's a facilitated diffusion protein, meaning a facilitated diffusion transport protein that moves the fructose into your cells from areas of high concentration to areas of low concentration. This we call facilitated diffusion, because the molecule is moving down its concentration gradient, but it requires the assistance of a protein. And this is the way fructose is digested. So once the fructose inside the cell equals the fructose outside the cell in your gut, no further fructose will move into the cell, meaning you cannot digest all of the fructose that you eat. At least some of it will never be brought into the cell because the fructose concentration will move into the cell only when it is higher outside the cell than inside the cell. Once the two concentrations of fructose equal each other, inside the cell equals outside the cell, fructose will stop moving into the cell. And fructose is moving into the cell by facilitated diffusion. Osmosis is the third way a molecule can move by passive movement into the cell. Osmosis is the movement of water into the cell from areas of high concentration to areas of low concentration. But it's not quite simple diffusion because it's a solvent that is moving, not a solute. And then the water is moving across a membrane. So osmosis specifically requires a membrane, a concentration and gradient of water, and then the water moving across the membrane into the cell. Now, actually, you can get reverse osmosis where the water concentration is higher in the cell than outside the cell, in which case water will remove by reverse osmosis outside of the cell. Anyways, osmosis specifically refers only to water moving across a membrane. So it's a little bit more than simple diffusion, because simple diffusion does not have to involve a membrane. And an example of that, actually I've got it in the next picture. Simple diffusion is simply where molecules move from areas of high concentration to areas of low concentration. Like when we put a drop of dye in this water, the dye molecules have areas of high concentration right here, and they will move to areas of lower concentration in the water. Okay, that's simple diffusion. And that's the way oxygen and carbon dioxide move. Let's go back to the water. Osmosis. It's not a solid, it's a solvent moving across a selectively permeable membrane. As you saw here, we're having simple diffusion, not across the membrane. Although you can have like oxygen moving into the cell, so this will make the cell, and then oxygen moving into it. The movement of oxygen is going to happen by simple diffusion across the membrane, but simple diffusion does not require a membrane. That's the point. Osmosis does require a membrane where water, only water moves across the membrane. And then water only moves from areas of high concentration to areas of low concentration. And that we call osmosis. All right, where's facilitated diffusion? Here we go. This is showing you with glucose, but I don't want to use the word glucose because with glucose it's more complex than this picture is showing you. I'm going to talk about it with fructose, and then this picture works perfectly for facilitated diffusion. You ate high fruit diet, you have lots of fructose in your gut. The fructose moves into the cell by facilitated diffusion using a transporter protein, which is the same as a transport protein, but it's a facilitated diffusion protein. And the movement of this molecule is happening without using ATP. So the molecule is moving into the cell without the cell spending any energy. And then the fructose moves into the cell. And this fructose will move into the cell until the fructose inside the cell equals the concentration of the fructose outside the cell. And that's facilitated diffusion. A solid using a transporter protein to move across the membrane. And this costs the cell no energy because the molecule is moving down its concentration gradient. And that's the way fructose moves into your cells. Glucose doesn't quite move into your cell this way. It moves into your cell this way only when the concentration of the glucose is higher outside the cell than inside the cell. So glucose moves into the cell by facilitated diffusion only when the glucose is higher outside the cell than inside the cell. When glucose becomes equal outside the cell than inside the cell, then this molecule changes and stops acting as facilitated diffusion and then acts as active transport. And then this molecule will pump every one of the glucose molecules outside the cell into the cell until you have 0 outside the cell. So glucose will be totally digested. Fructose will not be totally digested. One reason why you should eat fructose instead of glucose. But anyways, this picture works for fructose. So I'd prefer you to learn it for fructose. I'm not ever going to quiz you on how glucose is brought into the cell. We talked about osmosis. When we're talking about water moving into the cell, it's important to realize that a bacterial cell can experience 3 kinds of osmotic solutions. There can be an isotonic osmotic solution, a hypotonic solution, or a hypertonic solution. Each of these terms is a relative term. So it's correct or best to state that the solution outside the cell was hypertonic relative to the solution inside the cell. Any question about any of that? All right, let's talk about isotonic solution. This is the easiest one to discuss. Isotonic is when the concentration of water outside the cell is equal to the concentration of water inside the cell. That means there will be no net movement of water into the cell. Why did I say there will be no net movement of water instead of that there will be no movement of water into the cell? Come on, somebody's got to know. Nobody's going to take a guess. You guys do need to participate, otherwise the class is kind of boring.
Can you repeat the question one more
Not
time?
active. Somebody said something. What?
Can you repeat the question one more time?
I said when the solution is isotonic, there will be no net movement of water inside into the cell. But why did I say no net movement of water? I didn't say there's no movement of water inside the cell.
Is that because the amount of water inside the cell and outside of the cell won't change?
Yes, it is. The amount of water inside the cell is equal to outside the cell. So for every water molecule moving into the cell, there will be an equal water molecule moving outside the cell. But the concept is there's no net movement of water either into or out of the cell when the solution is isotonic. The cells are happy when they're in isotonic solution because their cells are not either gaining water or losing water. Any questions about any of that? All right. Now it's important to realize that I'm talking about water and the movement of water either into or out of the cell, but the concentration of the tonicity is not defined by the concentration of the water. It's defined by the concentration of the solute. So when we say the solution is isotonic, we're literally meaning that the concentration of the solute molecules outside the cell are equal to the concentration of the solute molecules inside the cell. Any question about that? So the tonicity is defined by the concentration of the solute, not by the concentration of the water. But it's the water, in this case, which will be moving. And because it's isotonic, the concentration of the water inside the cell is equal to the concentration outside the cell. For hypotonic solutions, the solute concentration outside the cell is at lower concentration than the solute concentrations inside the cell. So this will mean there's more water outside the cell than there is water inside the cell. And an easy way to remember this is the concentration of the water is just always the reverse of the concentration of the solute. So there's less solute outside the cell. That means there's more water outside the cell than there is inside the cell. When the cell is in a hypotonic solution, there will be net movement of water into the cell. This will cause swelling of the plasma membrane up until the plasma membrane will swell up until it touches the cell membrane. Once the plasma membrane reaches the cell membrane, the cell wall, the plasma membrane cannot expand any further. And that will limit water moving into the cell, meaning no further water movement can move into the cell because the plasma membrane cannot expand any further. So there will be no further water moving into the cell when there's a cell wall. And that protects bacteria as well as algae and plants from dying when they're put in a hypotonic solution. For animal cells, if you ever put red blood cells in a beaker of water, the water will continue moving into the cell and the plasma membrane will continue to expand up until the plasma membrane bursts. And then the cytoplasm will leak out of the cell, causing the cell to die. And all animal cells in a hypotonic solution will die. But not bacteria or plant cells that have a cell wall. And that's why you can find algae and plants growing in water, because they have a cell wall that limits the movement of water into the cell in a hypotonic solution. This is actually how penicillin G and the enzyme lysozyme can damage bacteria. They damage the cell wall, and then the water can move into the cell, and then the cell membrane can come out of the hole in the peptidoglycan and then protrude out, and then more and more water can enter the cell until the plasma membrane bursts by osmotic lysis. Any question about any of that? A hypertonic solution is when the concentration of the solute molecules outside the cell are greater outside the cell than they are inside the cell. When this happens, there's a net movement of water from inside the cell to outside the cell. So water will leave the cell. That will cause the plasma membrane to pull away from the cell membrane, and the plasma membrane will shrink, causing there's a name for that, and I'm drawing a blank now. Pinocytosis? That's what happens with the wilting of a plant cell, where water will leave the cell, and then the cells will shrink by pinocytosis, and that can then lead to death of the cell. Generally, if the cell is susceptible to water loss, which is most bacteria, like E. coli, the bacteria will then lose too much water, and then the cell will die. That's actually what happens with a bottle of molasses. My mother had a bottle of molasses that we figured was over 30 years old. And why it was still good was whenever we opened it and bacteria fell in, the bacteria would lose water because the bacteria were in a hypertonic solution, and then the bacteria would die. And that kept the molasses good for over 30 years. After 30 years, we finally decided, it probably would be a good thing to throw that bottle away. But at 20 years, the bacteria were still dying, and I made the molasses into molasses candy, which my father ate, and he loved it. So even after 20 years, the molasses was still good. And probably it was good after 30 years. We just never used it. And we decided, this is really old, we're going to throw it away. Tells you my mother never used molasses very much. And I only made molasses that one time, molasses candy that one time for my father. I think I was a hypoglycemic at that time because I didn't eat any of the candy. I don't particularly like molasses candy, so I wouldn't have eaten much anyways, but I don't remember eating any, so it's probably hypoglycemic. All right, so passive movement of materials is fine as long as the concentration of the material the cell wants is higher outside the cell than inside the cell. However, what's going to happen when a cell is in an environment with a low nutrient concentration, like a cell in the ocean or a cell in the desert? The molecules that the cell is going to want are generally going to be at very low concentration in the open ocean or in the desert. In this case, the cell cannot use a passive process for getting the molecules that it needs. And the cell will use an active process, such as active transport, to move the molecule that it wants against its concentration gradient, meaning move it from outside the cell at low concentration to moving it inside the cell where the molecule has a higher concentration. So the molecule is moving the nutrient molecules against their concentration gradient. This requires energy for the cell to do this. And generally the cell will use ATP to move the molecule into the cell. And it requires an active transport protein to pump the molecule from outside the cell to inside the cell. So this requires active transport, which requires a transport protein in the cell membrane, which generally is molecule specific, meaning for each molecule type, like glucose, that we want to move into the cell, we have to have one transport protein. And for another molecule, like the amino acid methionine, you have to have another transport protein to move methionine inside the cell. And then you require the use of ATP, which it'll be broken, and we get the energy from ATP to move the molecule against its concentration gradient. And this is active transport of the molecule. All right, any questions about any of that? I think we end at 6:50. Yep. So if there's no questions, I'm going to end it here, and we'll continue with this slide next time. Any questions? All right, if there's no questions, I'll see you at 7 for the lab.
Thank you.
All right.